Okay, welcome everybody to our next installment of the Kansi Ontario Stage International Seminar Series. Um, I This is sort of my favorite part of the stage program, and we love having investigators come from all over to talk about their research, and we're really lucky today with our current speaker. Stage operates with the support of Kansi Ontario, which is an EDU in the Faculty of Arts and Science at the University of Toronto. These seminar series are sponsored by lots of our partners here in Toronto, including the Hospital for Sick Children, Linnefeldt Tannenbaum Research Institute, Princess Margaret Cancer Center, U of T Department of Statistical Sciences, and today's seminar is sponsored by the McLaughlin Center. So before we get started, I'd like to do a land acknowledgement. Here we wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. And because we do have people online who might be in other parts of Canada, I encourage you to reflect on the privilege that it is to be able to work and live and play on this land. Okay, so I am very pleased to introduce our speaker for today, who is Heidi Rehm. She has a very long list of appointments. She's a very busy person, but among those, she's the director of the Genomic Medicine Unit at the Center for Genomic Medicine and Chief Genomics Officer, the first Chief Genomics Officer I've ever met, at MassGen, working to integrate genomics into medical practice. Among other things, she's the director of Broad Clinical Labs and is also the principal investigator of ClinGen. And this is another area of emphasis in her work, which is really sort of encouraging open science and providing free and publicly accessible resources to people who are studying genetics. So her passion really lies in the discovery of novel rare disease genes, and this is some of what she's going to be talking to us about today. So Heidi.
Thank you. It's a real pleasure to be back in Toronto, a place that's got similar weather to Boston. Okay, so I'm going to talk about the work that we do in rare disease. And just for those of you who don't spend all your time like I do thinking about rare diseases, just a reminder that although rare diseases individually are rare, collectively they are common, with 1 in 10 in Americans or 30 million in the US, 400 million globally that are affected by rare diseases. So it's a huge burden in terms of all countries in the world and these disorders. I particularly like this figure that the Orphanet group developed and published in a paper now 5 years ago, thinking about the challenges in rare disease. So to date, if you go to the OMIM database that tracks new discoveries in rare disease, there's just under 5,000 genes that have been implicated in single gene diseases. But over 50% of the cases that we sequence and test in a clinical lab, we can't solve, or even in a research study. And there's actually data that suggests there's over 5,000 more genes to be discovered. The challenge here is that what this figure is showing you is the prevalence of rare diseases, in the sense that about 77 to 80% of people with rare disease have it due to a more common rare disease that's prevalent in 1 to 5 per 10,000. So most of the people walking around with rare disease actually have it due to a small number of more common rare diseases. The vast majority of rare diseases, this bucket of 3,000, represent 0.3 to 0.5% of people with rare diseases. So what that says is most of the rare diseases we've yet to find are incredibly rare and really difficult to solve. So that's just kind of a little bit of the challenge that we struggle with in this space. So we launched, and this was launched by Daniel MacArthur and myself a decade ago. Now Anne O'Donnell-Lurie and I run our Rare Genomes Project. And our goal is really to sort of build a large-scale, phenotypically diverse cohort, and we would engage the community and really make participation by patients easy. We would return results so we could return value to those patients, and then we would rapidly share the data and the methods that we're developing. So we've been working in this project now for over a decade. This is our current team at the Broad Institute. And in addition to Anne and I, we collaborate closely with Mike Talkowski, Caitlin Samoha, and Monica Wojcik, and then have a number of members that are working on methods development, analysis, different technologies, long read, structural variant pipelines, all sorts of stuff. And I'll tell you about some of that work today. We also, as I pointed out, in terms of how rare diseases are, we collaborate with investigators all over the world, and some here in Toronto, and in all disease areas, and also with patient registries. So we're really trying to bring that. That's allowed us to amass over 20,000 samples through our collaborative efforts, and that's really critical for rare disease. So within the cohort that we've recruited within Boston in the US, we do it online so that we can recruit from anywhere in the US. Our IRB only lets us directly recruit in the US, but our collaborators can then recruit anywhere in the world where they are. But every blue dot is a family we've recruited. Every orange dot is a case we've solved and returned a result to. So we're really making a dent. Most of these patients have had clinical testing that was unsuccessful, but we've been able to find their diagnosis one way or another. And mostly through novel technologies and analytical approaches, but also a lot focused on novel data sharing initiatives. So, and by the way, feel free to interrupt with questions along the way. I don't mind at all. In fact, it's more interesting. So in terms of our progress to date, we've done, I wish there was a screen in the middle. We've done both exome and genome sequencing. Our strategy when you're dealing with large scale studies, cost effectively, we just exome everything. And then the unsolved, because we find a lot of diagnoses that are just obvious things that were missed clinically, and we can catch a lot of them with exome, and then we reflex over to genome. And it's just for a large scale approach, that makes most sense. Whereas I would say, as a clinical diagnostic lab, you'd want to start with genome. That's sort of how I think about it. But to date, if we look at our entire cohort, that is, again, mostly patients who have not been solved through clinical testing. About 35% we've been able to find a diagnosis for, about 45% are negative, and then about 20% are inconclusive. So I'm actually going to talk about what we do in each of these categories in terms of trying to solve these cases. So those that are negative, I'm going to have to figure out where to stand here. Those that are negative, it's either because the variant is missing from the sequencing data, and that certainly when we start with exome, it just may be in the non-coding region, right? So moving to genome is the best strategy there. Or it could be the variant is present in the sequencing, but it's overlooked. So in terms of addressing the missing from sequencing, we reflex to genome, and we recently published a paper looking at the true added yield of genome over exome. Now, lots of people publish papers moving to genome and huge yields, but what they really need to do is go back and say, could I have found that with the best in class current exome, which is what we did in this study. We actually relooked at, could this be found in our current exome with our current exome pipeline, and then really focused only on things that absolutely required genome. And that it was a 8.3% yield. And most of that is in structural variants, regions of the exome with poor coverage, deep intronic variants, and repeat expansions. Those are the biggest areas where the added yield from genome was demonstrable. Now, interesting, yes, question?
Variants. Is that mostly like splicing variants?
Yeah, so in this case, they're actually the added value of Splice AI as a predictor tool has been in our hands quite impressive. And so being able to predict a cryptic splice site deep intronically that's going to alter splicing, and then use RNA-seq or a mini assay to then validate that, we've been able to pick up a lot of solves through deep intronic data. And I actually have a slide on that in a second. Now, interesting, right after we published this paper, the discovery of RNU4 came out, which has been just impressive. It's the single most common cause of neurodevelopmental disorders, despite being an incredibly small gene with almost all of the variants due to one particular insertion. So that we and many others around the world were part of this collaborative study. And then that has since implicated a number of other non-coding RNA genes, largely that have been not put into exome capture kits. And so they've been sort of missed from this, but present in genome. And so if you update that slide I just showed you and add in just the non-coding RNA genes, our yield is now 9.4%. So a pretty impressive addition. I think none of us thought there was going to be a big yield gene out there in this stage based on that orphanet figure I showed you, but capturing non-coding RNA genes has proved us wrong there. It's a pretty impressive yield. We also had a side study that Ben Weisberg led looking at all of our cases with muscle disease, or actually all of the cases, but looking for SMA, which is a little trickier to pick up on exome, but you can easily find it with the right algorithm. And the number of cases misdiagnosed with different disorders, not spinal muscular atrophy, but misdiagnosed with limb-girdle muscular dystrophy, other things, is impressive. And all of these are patients picked up with a single known variant for which there's a treatment, yet all these patients not getting access to that because they had panel tests that didn't include SMA. So just showing you the added benefit of a genotype-first approach to diagnosis. So now coming back to the other side of variant missing from sequencing. So obviously moving from exome and genome, that's obvious. But the one question is how much better long read sequencing is capturing variation that is missed in short read sequencing. So we've been doing that along with many others. An analysis of 96 samples that we did by long read, we solved 15 out of that 96. This is a common thing that lots of people stand up and say, wow, like lots of added yield. However, doing the same thing we did comparing genome and exome, the question is, would a current best-in-class short-read genome with current pipelines have picked up those diagnoses? And the answer is 14 of the 15, yes. And they're in the area of new gene discovery has come out since that genome was run, or new variant evidence has come out since the genome was run, or just better SV pipelines picking up short-read structural variants. So there's really only one, and that was because it was a mosaic variant with very few reads, which we're just a little more confident with the long read data than what we picked up on short read, small fragments. So, and actually Alba Sanchez, who's a graduate student in Mike's group, did an analysis of 326 papers reporting long read data, and then looking for ones that actually took the time to look at short read data alongside, and their actual yield requiring long read was similar to ours, which is 1%. There's 0.6 and 2 from these two papers that you could actually look at the short read. So there's been a huge amount of hype that long read is, everybody needs to move right now to long read to solve all these cases. My comment is not for the price, because and the stability of the pipelines. There's not robust pipelines right now for analyzing this data. I think eventually we will get there as a community. The prices will come down and we will have better pipelines to analyze this data, and also better population frequency data from the areas of the genome that we don't have population data from, to be able to actually more productively look at this data in the regions that you're actually getting that you can't get from short read. So I still think there's promise eventually, but I can tell you we are not moving as the first line test to long read based on this kind of analysis. Okay, now the other sort of area of the negatives is the variant's there, but we just can't recognize it as causal, right? That's probably the bulk of these. So one of the things that we've been doing is focusing on adding RNA-seq to our pipelines, that we can pull out variants that are actually disrupting transcription. And so this is just a couple of cases. Some of these are pretty straightforward. Patient with myopathy, that there was actually clinical suspicion for a specific gene, but there was no obvious variance in that gene when it was sequenced. We did a trio genome with RNA-seq on muscle tissue. That particular gene was an incredible outlier in expression, very clear, and then we could see the altered splicing, exon skipping, and then we could look into that region and find a retrotransposon line 1 element inserted, which was actually mosaic in the mother. So very, once you have this RNA-seq data, can really help you interpret your genome much better. This was a little more challenging case. This was another case, and there were lots of genes underexpressed and overexpressed. And we sort of dug into all these genes looking for variants, couldn't find anything. We looked at intron retention, and this particular sample was off the charts of intron retention. And you can see in that space here, certain introns were being retained, but not all of the introns. So what was going on? So it eventually led us to looking at one of these RNA genes, RNU4A TAC, that's part of the U12-dependent minor spliceosome. This minor spliceosome complex facilitates splicing for about 0.5% of introns across our gene set. And so that's what's being disrupted here. And that's leading to a subset of genes showing over and under expression. We assume the overexpression is the added signal from the intron retention, and the underexpression is rapid degradation of certain transcripts because of the intron retention. So it's seeing both things both ways. And ultimately, we found one known pathogenic and one novel variant in this gene, and the phenotype was a very strong match. So with a little extra effort there. Overall, in our hands, we've been able to solve 14% of unsolved cases by adding RNA-seq. Now, I will say these are all muscle diseases. It makes a difference if you have access to tissue that's reflective of the biology. So you can get muscle biopsies pretty easily. Obviously, neurodevelopmental disorders, retinal disorders, hearing disorders, it's harder to get relevant tissue. So when you have relevant tissue and those genes are expressed, it's going to be a higher yield, but trying to pick up these things from blood and non-relevant tissues is a little more challenging. However, if we go back to the same data set, the biggest yield that we've seen yet to date is just reanalyzing the data. Just the rapid advances and new discoveries, both at the gene level and variant level and pipeline level, is really the most impressive thing than adding new technologies. So we've been focused on, the problem is, we used to reanalyze every case every year that was unsolved. But as the case load got larger and larger, our analysts' ability to stay up with that became challenging now that we have thousands of cases. So we worked through a collaboration with both Microsoft and Dan MacArthur's group in Australia and developed an automated pipeline, which we call Talos. It's been posted as a preprint, just submitted the revision, should be fully published soon. But we basically, through the Australian cohort and our rare genomes cohort, were able to validate the detection of causal variants from this pipeline. It actually could pick up 86% of our previously solved cases. And now being applied prospectively, it's picking up about 5% of new cases. And we sort of set this at a level that made it pretty optimizing for specificity instead of sensitivity, right? Because if it's very sensitive and gives you tons of stuff to look at, that doesn't help with the automation and the whole goal of reducing the labor. So we set it to be pretty specific. It picks up only about one variant per 200 cases per month. So that's pretty low amount of work to sift through. But it's finding new things in an automated way that we don't have to do that work as much across the cases. So that we're now building that standalone pipeline, which is publicly available, into our Seeker platform. And we're working on a project to really start to bring clinical lab data directly into our pipelines on the NHGRI-funded Anvil platform. And that data can then sit there and be subject to automated reanalysis for solving clinical cases over time. So we're really starting to focus on not research recruitment and new sequencing, but just hooking our pipeline up directly of the output of clinical lab data. So that's one of our focuses right now. So I've told you about genome, long read, RNA-seq, reanalysis, sort of what we do with our negative cases. But there's this bucket here of the inconclusive results that are kind of a bane of everybody's existence, the dreaded VUS. And so the VUSs that we find kind of are in two categories. Either they're in a novel gene, in which case our first attack is Matchmaker Exchange, and I'll talk a little bit about that platform. So we launched this federated network. It's one of the first federated genomic data sharing platforms that we launched now a decade ago. And it was in an era where nobody wanted to share their candidate genes because they really wanted that first author publication of a new gene discovery. But you just couldn't publish if you had one case, right? And based on that first slide I showed you, or second, whatever it was, based on most of these rare diseases being fewer than 1 in a million, no single lab researcher will ever have a second case, right? So the only way to solve these rare diseases is to find someone else in the world that has the same candidate. And so we basically said, okay, let's set up a system that you can put your candidate out there and be matched with somebody else who has the same candidate, but nobody discovers your candidate unless they have the same data you have. And so the way this works is you put your gene in your own node, and lots of people use Gene Matcher because anybody can just register a username and go in and put your gene in there if you don't have your own node. We have our own node at the Broad through our Seeker platform, so a lot of people that use our Seeker platform, which is free, can use that node. But either way, you put your gene in any one node, it queries all of the others, and all this data is sitting on each lab's own platform. You don't have to centrally share it. It's truly a federated network. And then if there's a match, the two groups get CC'd on a joint email with both groups involved. They see each other immediately, and then they can collaborate to exchange more detail on the phenotype of those patients, and is this a good match? To date, we've submitted over 1,500 genes. We've gotten 500,000 electronic match requests. It's triggered 13,000 matches to candidates in Seeker, and this has been the single most productive pipeline for novel gene discovery with over 300 novel genes, almost every single one benefiting from matches in Matchmaker Exchange. So from my end, and this is data from the whole platform, there's 15,000 candidate genes from 200,000 individuals from researchers all around the world in this system. So an incredibly powerful system for novel gene discovery. The challenge is, and we've actually now, through the Gregor Consortium, put out guidance to really encourage clinical labs to start using this, and actually some already do. GeneDx, which is one of the major exome sequencing labs in the world, they are the largest user of Matchmaker Exchange in terms of cases in Matchmaker Exchange. So we're now really encouraging all clinical labs to do this, but also not just because the problem with Matchmaker Exchange, it was both the solution early on to get people to share these candidates to make them not viewable to the public, but it doesn't engage functional researchers who want to make animal models of these genes and other ways that these candidates could actually, especially if you put your candidate in and get no match, it's just going to sit there, right? So we encourage people both to submit those variants into ClinVar, but note the gene is not yet implicated in disease, so it's a VUS, but it's at least in the public domain. And then we've also launched this new database, it's now a few years old, GenCC, which is I like to call it the ClinVar for genes. So ClinVar, you submit variants and your interpretation of those variants. In GenCC, you submit genes and your interpretation of a gene-disease relationship and the strength of the evidence, using essentially a framework that ClinGen developed to quantitatively score evidence for gene-disease relationships. So we now have 25,000 submissions on over 5,000 unique genes from 16 submitters that are all putting this data and classifying these gene-disease relationships. And about 22% of them are deemed insufficient evidence despite being published in the literature as novel gene discovery. So buyer beware of the published literature on these genes. This is just a summary of all the groups that are submitting to GenCC. So we initially launched this collaboration to get harmony across the public resources like OMIM and Orphanet and ClinGen, G2P, the Genomics England and PanelApp systems in Australia and England. But we also then found that all these clinical labs had their own repositories of curated gene-disease relationships to inform panel test development. And lots of labs were willing to share their curated data. And now we've been engaging research groups that have candidate genes to publicly put them in as well. They're all listed as limited evidence, but so those groups are putting both their known genes as well as candidates into this database. So we think it's a really powerful now publicly accessible database of pretty much covering all gene-disease relationships for Mendelian disorders. So that's kind of where we are with both the novel genes. Now, when you find a VUS in a known gene, you can't put it into Matchmaker Exchange. It's only designed for novel gene candidates. So then now, like, what do you do with all these VUSs and all of the known genes? So the different strategies and data sharing we're trying to sort of support to get to try to interpret VUS in known genes. And I'll just point out some data that we published a couple years ago. We worked with labs in the US and Canada, actually Christian's data from Sick Kids is in here, and basically showed that of 1.5 million tests run over a 2-year period for all the labs in North America, 30% of all genetic tests run for diagnostic purposes in symptomatic patients ended in a VUS. So a huge negative impact to patients, physicians, and the healthcare system. Of course, that rate went up as the panel size got bigger. And almost half of all variants in ClinVar are variants of uncertain significance. So it's a huge burden. Now, tackling this VUS problem, Doug Fowler and I wrote an article last year with 4 strategies to really try to tackle the VUS problem. 2 of them are really improved predictors of variant impact and scaling functional assessment, which I won't talk about today. But 2 areas I will mention briefly are improving standards for variant classification. And I'll show you how that actually can make a dent in the VUS problem, and sharing data. So one is around sharing population level data. So all of us that do genomic analysis for rare disease, the first step we do is use population data frequency from databases like Nomad to get rid of common variants that can't be rare disease causal. The problem is that most of that population data is from people of European ancestry, the big blue blobs across these different data sets. And so it's usually other populations and allele frequencies that rise separately in those populations to a higher level that can help you rule out the variation more effectively. So we really need more diverse data. And just as an example, this variant is essentially absent or incredibly rare in the Americas, but it's in almost a third of the population in South Africa. So if I had South African data, I can quickly rule that variant out because of somebody else's data, right? So we really need data from all around the world to be most effective at getting rid of a lot of variation from our eyes. And interestingly, when we moved from Nomad V2 to V4, which is the bulk of the data, what we showed is the sample growth was pretty large. That was in part because we incorporated the UK Biobank data into Nomad. But most of the UK Biobank, no surprise, is European ancestry data. So most of our growth was in European data, which is the light blue bar. Now, if you look at the number of variants in Nomad with an allele frequency over 0.1%, which we can pretty much therefore rule out as rare disease causal, almost no move in the fraction of variants in European data. But with just the small increase in non-European ancestry data, pretty significant increase in the number of variants more common, above 0.1%. So basically what this is telling you is that we've largely saturated our understanding of common variation in European ancestry individuals. We have not closely tapped it in non-European ancestry individuals. So just proving the point that we need diverse data here. So a focus of Nomad has been trying to get diverse data. The problem to date is that to make Nomad, cost effectively, we've used data that's been called according to the same pipeline at the Broad Institute. But that means it has to have been sequenced at the Broad, which obviously restricts the diversity of data. So in Nomad V5, we are actually the All of Us research program has intentionally recruited diverse individuals. And most of that data was sequenced at the Broad or processed at the Broad. And so we're incorporating that into Nomad V5, which will be coming out in the next few months. and that will add a bunch of diversity, but still diversity only within United States diversity. So our big focus now is a project we call Federated Nomad. And here, unlike to date where the data had to physically come to the road for our team to process, we are now working with sites around the world, including Gordon Lerner Ellis, I think is online, who's leading our Canadian collaboration. So each site is processing their data according to Nomad's pipelines and then coming up with a final aggregate allele frequency data set. And then that is no longer individual level data. And so it can be shared with us to put into Nomad. And so that is a project that we've been working on. We have monthly calls with all the groups around the world. We've been working through the Global Alliance to engage stakeholders all around the world in this project and the common standards that we use. And this is now a list which is on our website of all the groups that have committed to processing their data and including it in Nomad. And we also allowed them each to develop their own logo and have their own browser. And so the one that Jordan designed for Canada is here with a nice red Canadian leaf. So that'll be coming to all of you soon. So this has been a fun project, and we don't yet have the first data set incorporated, but I'm hopeful that by the end of the year or early next year, we'll start having this federated nomad approach working in place. Okay, my last bit of this talk before we hopefully will have lots of discussion is to talk about my next variant data sharing project. So I talked to you about Matchmaker Exchange, where two people put in their candidate genes and get matched. So we call that two-sided matching. You only get a match with two groups have the same thing and are brought together. The problem is, it only works for novel gene discovery. So if you've got that VUS in a known gene, you have no system to use. Or if you put it in and you get no match, it's like, well, somebody's probably got a variant in a case somewhere that they haven't noticed, right? So wouldn't it be nice if you could take your variant and query the world's genomic data and find your variant in somebody out there and get their phenotype back? That would be glorious, right? So that is what we call one-sided matching, and that is what we are working on. So some early examples of this, the same group that leads Gene Matcher for Matchmaker Exchange also launched Variant Matchers led by Nara Sabrera. And so she would just let people log into her database that contained all of their variants from their genome and exomes. And so this is me logging in, putting my case and my variant and getting back a list of 8 cases that had that variant with their phenotype. And so 4 of them were parents that were unaffected and 4 had various different phenotypes. I quickly ruled out this variant actually in my case. So sometimes the variant matching tells you you've got the wrong candidate. That's useful too. We have now, since our Seeker platform that lots of groups all over the world use because we make it free, there's now 70,000 genomes and exomes in it from all of the collaborators. And we got them to not opt out. So everybody is letting their data be queried and seen if they have a variant. And so basically, when you're in Seeker and you're looking, in fact, I think the next slide shows this, you're in Seeker and you're filtering your genome and you have a variant candidate in your case with your phenotype. Below this line is then any other case from any collaborator who's using the platform that hasn't opted out, which no one has. And their data, the quality metrics, the phenotype, and then you can click the blue button. It auto drafts an email for you, which includes your phenotype and saying you would like to collaborate, and then you send that, and then you can get more deeper details about that particular case. Actually, we've had our first gene discovery from variant level matching. There were 5 families that all had the exact same variant for a retinal disorder. So that was fun too. So we really think that this will be a way to open up data sharing in a federated way where no one has to centralize their actual raw data. And so we're now, through the Global Alliance, launching using a standardized API developed by the Global Alliance called the Beacon version 2 API that defines the various fields that we're sharing across these federated databases. We will allow maidenly focus on rare disease data sets to be able to query all around the world. So to date, we've now launched and connected 4 different databases: Variant Matcher, the commercial Franklin by Genox, which is used by thousands of clinical labs, Geno2MP and MyGene2 at University of Washington, and our Seeker database at the Broad. We've approved, so we have a steering committee because this is sharing individual level data through queries. We approve each person, and we have an authentication mechanism. It actually uses an OAuth protocol by Auth0, so that the only people that can query this use that authentication mechanism. Then we can, and the reason that's really important, we actually had to debate about just making a public API versus requiring authentication, is because we think more groups will actually share their data if they can understand who's querying it and set limits if they choose to. So any group that's sharing their data can reject any other group that they don't want to query. So far, everybody just opts into everybody, and we try to make sure there's no bad players here. But particularly when we get to getting the clinical labs to share their data, I anticipate that they're going to only share with others that agree to share back. And that reciprocal sharing is another reason why this sort of approved and authentication approach is important, because it's great if you have a platform and anybody can use it, but if that means they don't take the time to share their data reciprocally, then that diminishes the value. So we will probably make this a share and share alike platform. But researchers who want to come in to add value in other ways, we don't want to inhibit that. So we want researchers to come in, make as many functional models of anything in there as possible. We don't want to inhibit that. But groups that have data, we want to make sure they're sharing that data. So this way we can sort of approve and encourage. And there's lots of groups that are we have 2 monthly meetings with all these groups as we develop this system to share data. And this is just an example of how you see it in the system. So the idea here is, like, each group can let people query their database, but then you got to create an UI, create a login process, all of that. And nobody wants to log into 1,000 databases one at a time to find a variant. So here, somebody logged into Franklin, immediately sees whether the variant is in Variant Matcher. That's a real-time API query that's happening behind the scenes. This one isn't there, this one is there, and you immediately get the aggregate data. 726 times, 144 of which were homozygous. Click on that hyperlink, it lands you in Variant Matcher, you log in and get the phenotype. Now, if I was a rare disease clinician, researcher, I would look at that and be like, that's not a causal variant, move on. But sometimes it'll be like, one time, 0 homozygotes, and you're like, interesting. Let me go find that one case. So really fun. This is now our Seeker interface, where now you're seeing the results from MyGene and Variant Matcher here immediately, the het and homozygotes. You click on the blue hyperlink, it lands you in a database, you get the phenotype. So right now, it's mostly you see the aggregate het homozygote stats, click on a URL and go to the database. We're now working to just directly deliver the phenotype into the other side. So it's one step fewer for anybody who is willing to do that. That way, the group doesn't also have to maintain a website to get the data and log in and things like that. So both will be supported going forward. So then lastly is the sort of thinking about how standards for variant classification can actually address our VUS problem. So I've been part of the sort of professional variant classification standards development for a while now. We're in the SVC, Standard Sequence Variant Classification version 4 phase, which we're actually piloting it right now. We're just about to finish in the next week, phase 2 of the pilot, which is what's informing the final version of the guidance. We've really overhauled the whole system from the R2015 version to really reduce double counting of evidence and make it more quantitative. It's mapped to a Bayesian framework now. And then we've also introduced VUS subclasses. And I'll just point to a couple papers, one from the Invitae group and one that we published. And one of the take-home messages in looking at variants and how often they're reclassified and what direction of reclassification they go, what Invitae saw from 2 million variants over 8 years was that 79% of VUS went to benign or likely benign. In our paper, which was focused on labs that were already using VUS subclasses, which was Mass General Brigham, the lab I used to direct, the Baylor lab, LabCorp, and Quest. That was 150,000 variants. Again, 80% of US reclassifications went to benign or like benign. So clearly, our VUS sort of field is probably too much, including too many variants that are actually going to be benign. So that lesson, yes, go ahead. Yes. So if it was classified as pathogenic and solved, but then somehow got turned around, that's in this data set too, yes. That doesn't happen often, but it does absolutely happen. And in fact, this is deeper data answering exactly that question. So this was the 4 labs that use subclasses of VUS and have been using them for a decade or more. And so for each class path, likely path, VUS high, mid, and low, likely benign, you can see the direction of movement and the rate of reclassification of that category. So what you will see if you focus in on these VUS subclasses is that for the VUS low, no variant ever moved to path or likely path from that VUS low category. In contrast, that VUS high category, almost half of those variants moved to likely path or path. They were also, that category was the highest to have movement at all. 10% of those variants got reclassified on the VUS high. So it just goes to show you that doing this subclassification tells you a lot about what's going to happen to that variant. And that can help guide clinicians and how much energy to focus on the VUS. Yes.
The variant that would be like on the boundary between VUS high and likely pathogenic, or like on the boundary between
Yes.
VUS and likely benign.
Was there a question or?
Yeah, like you have an example of kind of like.
Oh, an example. Yes. So, I mean, each variant is going to have different amounts of data that are contributing, and things like observed in cases, or segregating, or functional assay, or de novo occurrence. These are all types of evidence. And for each piece of evidence, there's some like scoring. The current framework uses supporting, moderate, and strong, and very strong. And then there's like a rubric of 1 strong and 4 supporting equals this. And so in the framework we're working on now, it's points, 1 point for this, 2 points for that, based on the strength of that evidence. So it's just a question of how many points or pieces of evidence you've gotten. And if you're just, one more case might have put you over the line, right? Or a de novo occurrence, or a functional assay. So there's lots of variants. What's mostly happening on the VUS low to likely benign category is that the variant, like there's, 78% of variants in ClinVar have been submitted by one lab, despite there being 6 million submissions. And that is because most variation is rare and unique to an individual and never seen again. So these variants that we find all the time, you find it once, it's not in Nomad, and it's just sitting there with no evidence on it. Those are these VUS low. They're not high enough allele frequency in Nomad to rule them out as benign, but you have no data. So that is a classic variant. And then some new population data comes out, Nomad gets a new version. Now you have enough allele frequency data to push it to likely benign. That's that lower bucket and what's happening. Yep.
Have like, let's say like a variant with like, really high like missense pathogenicity scores.
Yes.
In a gene that's known to be pathogenic, but the variant only appears once.
Yep.
Where would that?
Right. So, and that's actually, I don't think I have the data from that. It's in a different talk. But so when we first came out with the SVCV3 framework, which wasn't called that, but the 2015 standards, we basically said these predictors could only give you supporting evidence, very limited evidence. However, those predictors have actually gotten better over time. And so now in the new calibration of some of the newer predictors like Revel and others, that they can actually score in the strong level of evidence, some of them. And so now this freaked a lot of people out in the clinical lab. We're like, what? These predictors suck. How can you let a variant get up with no other evidence? And actually led my colleague Anne O'Donnell-Luria and a postdoc Sarah Stenton to actually look how many missense variants in the genome on an average individual reach strong. And it's on average, at least in disease-causing genes, 1 per genome reaches that level. So it's actually very few variants with these better predictors are getting to that level. So it is now at a level that you could potentially get to close to likely path with certain missense variants and certain, if they really, but very few of them get to that level. Does that sort of address what you were talking about? But I will say it's easier to get them to likely benign with just a predictor. And we did that intentionally. So in the old framework, we sort of started with the premise, everything's smack in the middle of VUS, and then you got to get evidence to send it either way. The new framework is basically arguing most variants start here at the lower end, and then you move them one way or the other. And we've made it much easier to get to likely benign based on the data I showed you, basically. And in fact, a colleague in our hearing loss expert panel, Hila Aziz's lab, they took 179 VUS, they were all missense VUS, applied our draft framework and asked where they ended up. And basically what you can see is that half of them now move to likely benign and benign with the new framework. Another 30% are in the VUS low bucket. And if that's labeled appropriately on a report, basically 80% of these VUS reports, a clinician can say, okay, I'm just going to ignore this stuff, right? And then we also allowed stronger pieces of evidence to have higher scores. And so other variants are, and this comes to not just the predictors, but data like de novo occurrence and other things, there is strong evidence to move a variant up higher than it used to be able to. It used to be the framework was designed where no single piece type of evidence could get you any end of the spectrum. Now we're like, no, if there's been 20 de novo occurrences, that's really good data. If it's a specific phenotype to gene match, let it score high, right? So, that's sort of some of the framework changes that are going to influence things. I will caution to the thing we were talking about, which is the current framework allows more double counting. We have these other buckets like low rate of benign missense variation, active site, other things that are also built into these predictors. And so my advice to people is don't use these newly calibrated strengths of level if you're also adding a bunch of like indirect evidence of the other types that are just like, conserved region and this, that, and the other thing that are overlapping with these predictors evidence. So I think in the new framework, we're preventing that, but the current framework, it's a little hard to move that part of the puzzle. And we're also no longer allowing you to count absence and nomad as evidence in the new framework. But in the old framework, you could kind of get things to likely path with a lot of indirect evidence that's double counting. So buyer beware there. Okay, I'm going to stop in just a second. I think this is my second to last slide. So I will point out, interestingly, in that data on the rate of VUS in clinical lab reports, I left off one bucket, which was the rate of VUS in exome and genome reports, which is statistically significantly lower, which you could argue makes no sense, right? You find more VUS if you look at the entire genome and exome. So how could it possibly give a lower rate of VUS? Well, it's because it's a lower rate of what we put on reports. And if you use the labs that were using subclasses of VUS, multi-gene panel VUS, there's a quarter of them are these VUS low. Whereas in exome genome, no geneticist would ever put a VUS low on an exome or genome report because you're using your professional judgment to say, I'm going to put just variants that might be causal on this report. And in panel testing, you just interpret every variant and all VUS go on the report because often you don't even have the phenotype. So that is why there's a reduced rate of VUS in exome genome sequencing tests. I'll also point out from our other study, the VUS high bucket is also the smallest number of variants. So when you get one of these, we want physicians to pay attention. And we are also about to release guidance from ACMG on reporting VUS and how to deal with these new subclasses. So, key things being put the subclasses on the report. The UK has used subclasses for years, and they have fun terms like hot, cold, tepid, ice cold. but they only use them internally. They never share them with the clinicians. And that is not useful to helping guide the clinicians to do things with these variants. So with that, I'm going to stop there, leave the last 10 minutes for questions, and thank a lot of people that work on a lot of different projects that collectively advance our field. So thank you very much.
Might be questions online here, Alina?
While they're working on that, happy to answer any questions from the audience. Yes.
Thanks for the levels of evidence that enter into some of the scoring for the tools. What about the genotype phenotype one?
Mm-hmm.
Elaborate a little bit on how you gatekeep that, because that could get ugly.
Yeah, so like currently the description says it's called the PP4 line of evidence. Oh yes, the question was about the gene to phenotype specificity line of evidence. So for example, if I find a variant in the CFTR gene, just any novel missense change, whatever, it's in a CFTR gene, which is the cystic fibrosis gene, and the patient has an absolutely 100% clear diagnosis of cystic fibrosis, skin chloride test, like all the features of cystic fibrosis. The prior probability that any novel variant in that gene is pathogenic goes way up if that gene to phenotype specificity is there, right? However, if I sequence a patient with autism and find a de novo variant in Titin, which is implicated in both cardiac disease, dilated cardiomyopathy, and myopathy, and the patient has autism, like de novo evidence is a strong piece of evidence when there's a gene to phenotype match and you constrict the regions of the genome that you're looking at to a smaller number, just like a panel of genes associated with that disorder. So in fact, there was a study done looking at the Titin gene and autism, and they found 6 de novo variants. Why? Because Titin is the largest coding sequence in the genome. It's an incredibly large target. And on average, we have 1 de novo per coding region and about 100 per genome. So that phenotype specificity rule is really critical in terms of evidence. And so, for instance, when scoring de novo evidence, we actually require you to document the phenotype specificity. Now, the challenge is it's very subjective. Like, I can sit here and describe that, the patient has cystic fibrosis and they have a variant CFTR, and that kind of sounds right to you all, but like, how do I quantitate that, right? So in the new framework, and actually we've already published this paper through the ClinGen variant classification work group, we're using diagnostic yield of the phenotype as a quantitative metric for specificity. So if a patient with an elevated sweat chloride result gets cystic fibrosis testing, what is the positive yield for a diagnosis with known pathogenic variation for that phenotype and that gene? Let's say it's 80% of elevated sweat chloride leads to a positive CFTR result. Well, then that phenotype specificity can equate, and we have a point conversion of points based on diagnostic yield for that phenotype. And that's how we are quantitating the phenotype specificity, basically, if that makes sense. Yes.
I have a question about the role of proteomics in terms of providing additional interpretation of the splicing variants that may be subject to nonsense-mediated decay,
Yep.
may be irrelevant to biology.
Yeah, so this is an area we're just starting to delve into, is to see if we can get proteomic data to actually be an output for a given gene. We're just starting there. We don't have any data, so I can't really answer that question. I will say, having delved into the metabolomics area, which is not too different in terms of complexity and novel techniques, is incredibly noisy space and difficult to, unless you like know what you're looking for in advance, difficult to sort of see novel signal out of the background noise of this data. So I'm sure that there's a positive place in the world for this proteomics data, but I think it's going to probably go through some growing pains. And even RNA-seq data, largely one of the best uses of it is when you had a splice AI score, knew what gene to look at, then went into your RNA-seq data and looked at that gene, and then you could see. Whereas doing it genome-wide transcriptomic analysis was harder because of just the noise signal. So my guess is proteomics, and the same is true of metabolomics, starting will be like when you have a specific inference, a candidate or something, you want to kind of look into that data. And then over time, as we get better normal, control data sets to compare things to, and actually we're doing pretty large scale proteomics work with Olink for the All of Us data set with thousands of samples. As we generate these large scale data sets that become controls, it'll be easier then to look at case data and try to make sense of it. Does that help?
Thank you.
Question?
Joanna Roman, see on the screen. You
Ah.
can only see it on the screen. I don't know why. So-
Can you comment on concerns of VUS classification for different conditions, specifically how to interpret when a variant has distinct consequence on different conditions? Yes. So the most, well, not most, but at least a third of genes are associated with more than one condition. And so when we evaluate a variant, you need to consider it for all of the different conditions. So I gave the example of Titin. It's implicated in dominant dilated cardiomyopathy, but also implicated in recessive myopathy. So when we evaluate variants, there's definitely, we need to think about all of those conditions. So for example, there might be a condition where loss of function is the mechanism for this condition and gain of function for that condition. So the functional assays I might use to evaluate that protein will be different for those two. Also, the allele frequency threshold to call something benign will be a different number, a higher allele frequency necessary to call a recessive condition benign, or the variant for a recessive condition, versus a much lower allele frequency that I can use to say it's not going to cause the dilated version, right? So you really have to think about all of the conditions and what frameworks make sense, sort of what evidence types for each of those conditions. Now, sometimes for a lot of things, they kind of span both, and often, like in hearing loss, a lot of the genes cause both recessive and dominant. We'll just sort of use the recessive thresholds because they're more conservative than the dominant, and usually we can kind of quickly and easily classify for both conditions. But if you were really scrutinizing, then you could differentiate those thresholds and sometimes, and even the predictors. I think this is an area that people don't think about. Those predictors work better or worse for different genes. They also work better for worse for different mechanisms of pathogenicity. So if you're calibrating and validating a predictor, you want to make sure you're using all types of variation for all the different conditions, gain of function, loss of function, et cetera, because you may get different results. Hopefully that answered it.
One last on that line, what percentage of your case is C2 likely pathogenic or likely pathogenic? And along the same lines.
You mean in distinct genes or
Yeah.
in the same gene, distinct genes?
Related to this question, when you go back and look at those that you characterize as pathogenic, say on exome, you reclassify based on a secondary finding, a second one.
Okay, so the first question was, how often do we essentially see almost dual diagnoses? We have seen very few of those. I remember the first paper that came out from Baylor, and the rate that they saw was 5%. And I think there's a couple things there. I think the variant classification rigor has increased, and so probably not all of those cases were true dual. The other thing is early on, when exome and genome sequencing were just getting started, the cases that would be sent would be those really complex cases that you couldn't decide what phenotype panel to sequence, and they were more likely to be true complex dual diagnoses cases with like two different things going on that made it challenging to decide what panel to order. So I think now that most labs, are using very large panels or doing exome or genome, particularly for the complex phenotypes, I think we're seeing a lot fewer dual diagnoses. They definitely happen and are out there, but in our experience, been pretty low. Not to say there aren't going to be modifiers and today, because it's hard enough to get a primary diagnosis given the rarity of these diseases, but to have enough data to look for modifiers, which are undoubtedly there and are giving differences in severity and pleiotropy of the phenotype, like that's why we need data sharing, because we have to amass all these cases together if we're going to start to look for the modifiers, which are absolutely going to be in there.
Just can I ask one more question on behalf of the community? Your resources have been throughout the world. Have the NIH cuts acted yet or no? Simple answer.
No.
We need to, and is there anything we can do to help?
I appreciate that question. Absolutely. So this year I laid off a third of my lab. So it's absolutely having impact because of the NIH situation. So all of my funding comes from NHGRI, the National Human Genome Research Institute, because of the encouragement for all NIH employees to quit and retire, and early incentives to retire early. NHGRI lost 75% of its staff that processes grants. So they actually have money to award, and they don't have the staff to get the money out the door. And so like our grants, our carry forward requests have not been processed, like money just hasn't come in. And new RFAs like the Gregor Consortium, which is the NHGRI funded rare disease program. That RFA was supposed to come out last March. It never came out. It's still waiting to be approved by HHS, which is so NIH and our new director, Jay Bhattacharya, say he's the optimal director of NIH, but he's less of the problem than his boss, RFK, which has been a huge impact in getting any new funding out the door, new announcements. So the Gregor RFA never came out. We're still waiting for it. We're right now trying to apply for cost extension to just keep the Gregor sites alive until hopefully someday this RFA comes out and refunds the program. But even refunding it, it's at a lower level. So this has been a huge challenge, and our government was shut down for 2 months. Huge numbers of study sections were all canceled. So like our ClinGen grant that we submitted in September isn't going to be reviewed until April. And so now we're talking about a cost extension for ClinGen because we can't get money in time to refund the program. So like it's not a total disaster, but it's a half a disaster, right? So it's a huge problem. I definitely think, and I'm part of the GCBR, the Global Biocore Resource Consortium, whatever that is, or called, but it's recognizing that there are global resources put out by not just my group, but groups all around the world, and that are used by all around the world. And we all, as a global community, need to collectively support. So there's efforts, and actually the GCBR is now being taken in by EBI, the European Bioinformatics Institute, and starting to really think about how to get other countries to contribute to these critical resources, make sure they're not dependent on one country, things like that. So, and I work very closely with, I'm the chair of the Global Alliance. The headquarters are partly a host institute here in Toronto at OICR under Peter Goodhand. So, working very closely here with the Canadian Host Institute for Global Alliance to help contribute to global resources and standards. So there's already Canadian input, but I encourage anything you or anyone else can do to contribute to those efforts. So thanks for bringing that up.
So we're just a little bit over time. I'm going to thank Heidi for an amazing talk and answering all of our questions, and sort of close it down for those of you online. Those of you who are in the room, you can probably be able to squeeze in a few more questions with Heidi, and there's lunch and coffee and stuff at the back of the room. So
Great.
Thanks very much, Heidi.
Thank you.